So we'll see the first image. Okay, it's an image of a peripheral smear. Okay, so if you see here, you can see a lot of abnormal shaped cells. Okay, so one important characteristic abnormal cell that can be picked up in this peripheral smear is this one. This is looks abnormal. Okay, there's one more cell here. I'll put it in a different color. Okay, there's one more abnormal cell here. Okay, and these are also small cells which are also looks abnormal here. Okay, so there are many abnormal cells in this peripheral smear. Okay, one which is in the center, which I have noted with a red arrow, is a classical sickle cell. Okay, okay, followed by a cell which is marked here, both cells. Okay, then few small cells here. This purple color cell is could be a nucleated RBC. Okay, fine. So these are there are a few target cells also, you can see here. These are some few target cells. Some target cells. Okay, with central pallor. Okay, fine. So this is a classical picture of sickle cell anemia. Okay, sickle cell anemia. Characteristic sickle shaped RBCs in between the peripheral smear. Okay, so these are the abnormal cells you will see: sickle cells, both cells, target cells. Okay, and you will also see something called nucleated RBCs. Okay, okay. So this is a classical picture peripheral smear of sickle cell anemia. Okay. So what is the problem in why do the patients develop sickle cell anemia? It's all because of one single point mutation. Okay. So what is that point mutation leads to? Okay. So the cause for sickle cell anemia is point mutation. Okay. Point mutation causing a change in the amino acid. Okay. Glutamic acid is replaced by valine. Okay, so what polar glutamic acid is replaced by non-polar valine. Okay, so the whole a polar amino acid is replaced by a non-polar amino acid. So the polarity of the amino acid changed. Okay, so is that a problem? Yes, it is a problem. How? Let's see here. See, as I told you earlier, this happens at the 6th position of the beta globin chain. Okay, fine. So when this happens, okay, so what happens is this is a normal oxygenated hemoglobin. Okay, two alpha chains, two beta chains. Okay, when the hemoglobin gets deoxygenated, okay, what happens is it will expose a hydrophobic patch or hydrophobic site like this in a deoxygenated state. Okay, the sickle cell or sickled hemoglobin will generate something called hydrophobic sticky patch. Okay, there is something called hydrophobic sticky patch. Both HbA as well as HbS will have a complementary sticky patch. Okay, it's kind of a receptor to bind the hydrophobic sticky patch. However, they're exposed only in the deoxygenated state. Okay, so at low partial pressure of oxygen, deoxyhemoglobin S can polymerize together to form long insoluble fibers. Okay, this binding of deoxyhemoglobin A terminates this fiber polymerization. Okay, because hemoglobin A lacks the second sticky patch. Okay, necessary to bind another molecule. Okay, so this polymer is because of this hydrophobic sticky patch, which is produced because of the mutation in the amino acid. Okay, so this is the reason for sickling of the RBC. Okay, fine. Fine, so that's about it. Okay, so how do you identify the sickle cell anemia, or how to diagnose it? Okay, one is peripheral smear. The other important investigation that you can do is you can do something called high performance liquid chromatography. Okay, to look for hemoglobin variants. Okay, this will actually pick up the common variants, which includes hemoglobin S, thalassemia, okay, etc. Okay, fine. So that's about the techniques to identify sickle cell anemia. Okay, high performance liquid chromatography, which is commonly used nowadays to analyze hemoglobin variants, which includes hemoglobin S, A2, and hemoglobin F. Okay, which is reliable, sensitive, and automatable. Okay, but it is costly. Okay, hemoglobin erythropoiesis is also done, but it's only to diagnose the rare variants, not used obsolete. Okay, fine. So that's about this image. Okay, it's a peripheral smear showing a characteristic features of sickle cell anemia. Sickle cell anemia is caused due to a point mutation in the 6th position of the beta globin chain, the beta globin gene, where a polar glutamic acid is replaced by a non-polar variant. So because of this change, the hemoglobin S will generate a sticky patch. Okay, this sticky patch will be bound to this hydrophobic pocket to form a polymer like this. Okay, so this polymerization can be inhibited when an oxygenated hemoglobin A comes in, because it doesn't lack a receptor to bind the sticky patch. Okay, this is called sickling. Okay, so that's about sickle cell anemia. That's about this first image. Okay, so image 1 is on sickle cell anemia. So we know what is the cause, and we know what are the tests we can do. High performance liquid chromatography. Electrophoresis can be done, but it's only for the rare variants. Okay, not for the routine variants like hemoglobin S. Okay, this is our second image number 2. Okay, so this image is a characteristic feature of hemolytic anemia. Okay, so this is a characteristic picture of hemolytic anemia. Okay, anemia. So where you can see this classical cells called Heinz bodies. What you see here in the inset, Heinz bodies, and the cells will look like bitten off. Okay, these are the hemolytic cells. These are the hemolytic RBCs. Okay, fine. So hemolytic anemia can arise because of hemolysis, which can be intravascular hemolysis or extravascular hemolysis. Okay, the intravascular hemolysis is usually characterized by hemoglobinuria. Okay, causes are many. Okay, one of the classical common cause of hemolytic anemia related to biochemistry is G6PD deficiency, glucose 6 phosphate dehydrogenase deficiency. Okay, okay, why glucose 6 phosphate dehydrogenase deficiency causes hemolysis? Okay, we already discussed in the morning session. Okay, glucose is converted to 6 phosphogluconolactone. Okay, so that upon by phosphogluconolactone oxidase to form gluconate. Okay, okay, fine. During this first two steps, G6PD is catalyzed, the first step. Okay, G6PD. Okay, and NADPH produced, and next step also NADPH is produced. Okay, in this NADPH is very important for maintaining the RBC membrane integrity. RBC membrane integrity. Okay, fine. So what it is, for example, you see here, since RBC is constantly involved in gas exchange, There is carbon dioxide and oxygen. Okay, there's increased chance for this oxygen to get partially reduced, forming hydrogen peroxide. So RBCs are at constant risk of oxidative damage. Okay, which can ultimately damage the membrane, RBC membrane, leading to hemolysis. Leading to hemolysis. Okay, fine. So that's about this thing. So when that this is prevented by NADPH. Okay, so usually hydrogen peroxide is produced in the red blood cell should be converted to water. Okay, for this you need glutathione. Okay, glutathione is an antioxidant. Okay, made up of 3 amino acids: glutamic acid, cysteine, and glycine. Okay, this glutathione will reduce this hydrogen peroxide to form water. Okay, during this process, glutathione will become oxidized. Okay, and this oxidized glutathione should be converted back to reduced glutathione, for which we need NADPH. So if there is no NADPH, glutathione cannot be recycled back to oxidized glutathione. So glutathione levels will fall. If the glutathione levels fall, then in H2O2, hydrogen peroxide cannot be converted to water. So H2O2 will accumulate. What that will do, as I told you earlier, it will cause oxidative stress. It will cause hemolysis. Okay, so that's about it. So does patients with G6PD always present with hemolysis? No, the answer is no. They usually develop hemolysis only in the presence of certain triggering conditions. Okay, what are those conditions? Those conditions include intake of drugs like antibiotics, antimalarials, okay, infection, and intake of some beans called fava beans. Okay, these are the certain triggers for hemolysis in patients with G6PD deficiency. Okay, infection is the most common trigger. They will be absolutely normal. They will develop some infection. Okay, and they will be given antibiotics that will trigger thing, especially antimalarial drugs. Okay, fine. So that's about this G6PD deficiency. Okay, so NADPH, apart from having a role in maintaining the integrity of the RBC, NADPH is also essential for fatty acid synthesis, steroid hormone synthesis. Okay, so these are the other important functions of NADPH. Okay, there are various sources of NADPH in our body in addition to glucose 6-phosphate dehydrogenase. Okay, what are all the various sources? We know G6PD, isocitrate dehydrogenase, isoform 1. Okay, that also produces malic enzyme, cytosolic malate dehydrogenase. These are other forms of NADPH. Okay, NADPH is also very important for phagocytosis. Okay, it's also called as oxidative burst. Okay, phagocytosis. Okay, so coming back to the picture, there's a picture of hemolysis. Okay, showing Heinz bodies here. Okay, and there are some hemolytic RBCs seen here. Okay, so the classical condition that can cause hemolysis like this is G6PD deficiency. Okay, fine. We know how G6PD deficiency causes hemolysis. We just now explained it, which was in which glutathione plays a very important role. Okay, fine. Now the next figure is image number 3. Okay, image number 3. The this is actually a bone marrow picture. Okay, this is actually a bone marrow aspirate showing the characteristic cell with wrinkled tissue paper appearance. These cells are called as wrinkled tissue paper appearance. These cells are called as Gaucher's cells. Okay, as the name suggests, this diagnosis of this condition is Gaucher's disease. Okay, so what is this condition? This is actually a muco or sphingolipidosis. Okay, characterized by accumulation of glucocerebroside enzyme defect is glucocerebrosidase enzyme responsible for degrading the glucocerebrosides. So this enzyme is defective. What happens? Cerebrosides, especially the glucocerebrosides, will accumulate, leading to sphingolipid storage disorder or mucolipidosis. Okay, fine. So I told you earlier, it's an bone marrow aspirate showing the presence of cells with crumpled tissue paper appearance, also called as Gaucher's cells, characteristic of Gaucher's disease. Okay, fine. So there are various mucolipid storage disorders. Okay, like there is something called GM1 gangliosidosis, is caused due to the defect in the enzyme called GM1 beta galactosidase. So that GM1 ganglioside will accumulate, and this mostly involves bones. Okay, fine. The next one is Tay-Sachs disease, which is also called as GM2 gangliosidosis, caused due to the defect in the enzyme called hexosaminidase A. Okay, accumulation of GM2 gangliosides, and it causes blindness. Okay, Gaucher's disease, glucocerebrosidase, accumulation of glucocerebrosides, again erosion of long bones. Fabry's disease, alpha galactosidase, okay, skin involvement and kidney failure. Fabry's disease, ceramidase, ceramide bone involvement. Kraft's disease, galactocerebrosidase. Gaucher's is glucocerebrosidase. Kraft's is galactocerebrosidase. Okay, and that leads to mental retardation. Okay, metachromatic leukodystrophy is caused due to the defect in the enzyme called aryl sulfatase A. It causes accumulation of sulfatides. Okay, accumulation of again causes mental retardation. That's it. Okay, fine. So these are the various mucolipid storage disorders. Okay, Tay-Sachs, GM1, okay, Gaucher's, Fabry's, Fabry's, and Kraft's. Okay, and metachromatic leukodystrophy is caused due to the different enzyme called aryl sulfatase. Okay, fine. So in all these mucolipid storage disorders, okay, you can see from this table that it mostly causes bone involvement, mental retardation, or CNS involvement, are the characteristic features of this mucolipid storage disorders. Lipidosis. Okay. One additional point about this Gaucher's disease is sometimes to know the disease severity, we can estimate an enzyme called chitotriosidase. Chitotriosidase activity is measured to know about the disease severity, especially the Gaucher's disease. Okay, so that's about the Gaucher's disease. Okay, so coming back to that question again. Okay, so this is a image 3. Diagnosis is Gaucher's disease. They can ask you about the enzyme defective, or they can ask you about the enzyme which is used measured to assess the severity of the disease. Okay, and these cells are called as Gaucher's cells, cells with wrinkled tissue paper appearance. Okay, cells with wrinkled tissue paper appearance. These are called as Gaucher's cells. Okay,